The main male sexual response is going to be the erection. Now, I've mentioned before the erectile tissues in previous videos, but just to keep in mind again of how this is all working, the erection is based off of the blood filling these erectile tissues. So what happens is the blood engorges the erectile tissues, especially expansion of the corpora cavernosa and corpora spongiosum. Now, this is in response to sexual stimuli. That sexual stimuli could be a visual, could be a mental. There's different things that could be triggered and causing a sexual stimuli. It varies from person to person. But what's happening here is you're compressing the drainage veins. So both of those erectile tissues, as they fill and engorge with blood, all the arteries bringing blood in, the drainage veins get compressed and they can't take the blood away. So therefore, an increased amount of blood is remaining in the erectile tissues. And again, remember, corpus spongiosum is around the urethra. Its purpose is to keep the urethra open. Make sure the semen can pass through the urethra. Now, even though this is the response, it can be induced or inhibited based on emotional situations and higher brain activity. Now, for example, it can be inhibited if someone's in more of a depressed state. If they're depressed, feeling down, no matter what, they might not be able to get an erection to occur because their mental state is blocking that response. But if someone is simply just thinking about certain things that could basically trigger or induce a sexual response, might not be anything physical or visual around, it might be a pure mental state. So both inducing and inhibiting based on emotions and higher brain activity. So what we're seeing here is the top view is a flaccid penis, so basically not erect. The bottom view is going to be an erect penis. What you'll notice, look at this drainage vein up top. That vein is wide open, perfectly round. But then when you come to the erection view, that vein is being compressed. Not as much blood can be leaving through that vein, which means the erectile tissue you can see down here have more red. There's more blood that's filling the erectile tissue, and it's staying there because it's compressing the venous return. All right, that is the response, but the whole response, the sexual response, is meant to deliver sperm. That's the idea of the gametes. So how do we even create sperm? Well, you create that through spermatogenesis. Spermatogenesis is the process of creating the sperm, creating the haploid sperm. We'll get to that. Now, most of the cells in your body are diploid, 2N. That means you have two sets of chromosomes, basically 23 pairs of chromosomes. So a total of 46 chromosomes or 23 pairs. And that's almost every cell in your body. Almost. Gametes, in this case that we're talking about sperm, but it'll be the same thing for the eggs coming up later in the female system. So any gamete is going to be haploid. Haploid is 1N, not 2N. It means you have 23 chromosomes, not 23 pairs, just 23 chromosomes. That means you have half the amount of genetic code you need to make a normal cell. Which that makes sense because as the sperm combines with the egg, half and half, you end up making a whole. So by having half the genetic material from the sperm, half the genetic material from the egg, when they combine, you're back up to diploid, back up to the full 46 chromosomes, 23 pairs of chromosomes. So how does this work? We're looking at the process of meiosis. Now, hopefully meiosis rings a bell back from AP1 or back from previous biology classes. But meiosis and mitosis are the two main processes of cell division. Mitosis, one cell, makes two exact copies. They're all diploid. But meiosis, one cell makes four, and each one of those four is haploid. So we start with prophase 1. In prophase 1, there's an interesting feature that happens in meiosis. You go through crossing over. What that means is the chromosomes, they actually exchange genetic material. You can see right here, there's some green and purple that have swapped over. So you have this mixture of material. This is the principle of why every child from a couple isn't the exact same. They're all different. Yes, they're similar, but they have their own differences because of this crossing over. The actual chromosomes are exchanging material, which means there's always variation. Now, in metaphase, everything's lining up right along the middle, which brings us to anaphase. They start to split apart. Half goes one way, half goes the other way. So we're splitting these chromosomes up. And by telophase, we're now split into two sides. But if you notice, this is all prophase 1, metaphase 1, anaphase 1, telophase 1, because it's only the first half of meiosis. The second half here goes through prophase 2, metaphase 2, anaphase 2, telophase 2. You take those two cells you had originally at the end of meiosis 1, and they then branch apart the same way to create 4 by the time you're done. So the idea of 1 becomes 4. 1 diploid cell becomes 4 haploid cells. So if we put that into the sperm production, spermatogenesis, we're going to notice that up top, you have two types of cells, type A and type B. The type A cells, those are going to stay there. They're not going to leave. They are permanent. Those are going to be your spermatogonium. But they'll divide into this type B cell, this type B daughter cell. That's what will go through the rest of the process, which then goes down into primary spermatocyte. The primary spermatocyte then divides into the secondary spermatocyte. You're dividing. The secondary spermatocytes divide into the early spermatids. We're now down to 4. So we've gone 1, 2, 4 at this point. And those early spermatids, they're not sperm, go into late spermatids. Again, not sperm yet. Late spermatids. They're still in the membrane, they're still bound, they're not leaving yet. From there, then they'll move on to be released. And that's spermatozoa. Remember, they're non-motile yet. They can't move. That occurs later on in the process. This is just creating the mixture of genetic material that goes into each sperm eventually. So a different way to look at it. Same names, but we're putting the 2N or 1N. So is it diploid or is it haploid? You'll notice up top, spermatogonium 2N. That's the stem cell. It stays there. It keeps dividing over and over throughout life. But then via mitosis, it makes the primary spermatocyte. We're still at 2N. Now meiosis kicks in. The secondary spermatocyte. You divide into 2, but notice because meiosis, we're now down to 1N. We're haploid. So each of these two cells only has half the genetic material. From there, we go through meiosis 2, and now we've created the spermatids, 4 individual cells. They're all haploid again. There, we're getting down to the spermatozoa eventually. So the process of going through this whole entire production of sperm, from diploid 2N to haploid 1N, 23 chromosomes. Now what we're looking at here, this is going to be one single seminiferous tubule. What you're seeing here is all these in the middle, those are all the tails of the forming spermatozoa. The actual cells you can see out in the wall here. So it's kind of giving you an idea of how it moves from the very outside of the wall. As it progresses, it moves down towards the lumen, where eventually it's released and will travel out of the testes and then move on to the rest of the process. So a little bit about how sperm is being produced and how spermatogenesis is occurring.